So, okay. So, hello everyone. Welcome to our genomics and bioinformatics seminar today. And we are very glad to invite Dr. Ryan Collin to give us a talk today. So before I pass my microphone to Dr. Zoltan Zalasis from the BCH, I would like to introduce our RC Wiki page, Digital Science Education. So you can see from this web page, we have what upcoming seminars here, which are scheduled. And what the previous seminars actually are recorded here. So if you want to watch the previous talk, you can basically click the link and what the talk was recorded in the YouTube. And if you want to go to this page, basically you can type RC Wiki, and you can go to our research computing wiki page, and if you scroll down, you can find the digital science link. If you click that, you will find what are scheduled seminars. So with that, I will I think I will pass it to Dr. Jonathan.
Yeah.
Can you introduce our speaker today? Thank you.
Absolutely.
Thank you
so,
for joining us.
Yeah, so welcome everybody. I think we are in for both an exciting and very useful talk. So our speaker today is Ryan Collins. He is an advanced PhD student at the BIG program, that's the Bioinformatics and Integrative Genomics program at Harvard. And I think many of us will be really interested to hear his talk because he's going to talk about structural variants in germline sequencing, whole genome sequencing data as part of the NOMAD project. So without much further ado, I'll hand it over to Ryan. I just want to tell you that please send your questions. That's usually our way of asking questions. So please type and send your questions to the co-hosts. So at the end of the talk, we can ask Ryan your questions. Ryan.
Great. Thanks very much for the introduction and for the opportunity to present. I'm presenting on behalf of, as was mentioned, a large consortium of PIs and researchers located around the globe as part of the Genome Aggregation Database, or NOMAD, and I believe Nikki Whiffen, who's also one of the leading analysts in NOMAD, presented at a recent seminar. So many of you will probably have some background on NOMAD, but I'm also presenting on behalf of Mike Talkowski, my PI, who regrets that he can't be here today, but will present basically the same information. So what we're going to be talking about today, as was mentioned, is human germline genetic variation. And so canonically, in the simplest form, you can think of germline variation as individual nucleotide differences, or SNPs, or SNVs, or however you want to call them. But as I'm sure many of us on the call know, and as we've known for many decades, genetic variation is actually far more complex than just single nucleotide changes. In fact, it's actually fairer, I think, to conceive of human genetic variation as a continuum of mutational size. And so, like I mentioned, we know quite a bit, and even a lot of the most canonical principles and first principles of genetics, and especially Mendelian genetics, were gleaned from short variants. So I'm referring to those less than 50 base pairs, which include single nucleotide changes, but also small indels on the order of 5 or 10 bases, or even 1 or 2 bases. And due to these variants being more readily captured and cataloged in populations, we actually now know that each genome harbors many millions of these short variants, and we also have large reference databases such as NOMAD, but before that, the Exome Aggregation Consortium, or EXAC, and many other data sets like the 1,000 Genomes, and even going back to HapMap, that have cataloged these short variants at scale across populations. The flip side of that coin is that we've also known actually for close to 70 years now that our chromosomes are not fixed in size and shape, and actually with surprising frequency rearrange and either gain or lose or otherwise structurally mutate. And based on cytogenetic and molecular and microscopic methods going back many decades, these variants were observed preferentially in severely affected individuals and tumor genomes, but have been observed very large variants at appreciable frequency in the general population. But actually, you'll notice here that there's an entire middle piece of this slide that's missing. And so this is what is encompassed by structural variation. Everything greater than 50 base pairs of change against the reference genome, and includes everything up to whole chromosome cytogenetic abnormalities. But actually, there's a ton of variation in this middle space on the order of hundreds or thousands of base pairs, which includes not only just copy number variants like deletions or duplications, but also balanced and complex rearrangements like short sequence insertions, inversions, reciprocal translocations, and like I mentioned, other more complex rearrangements, which I'll talk about a little bit later. But the real problem here is that we know that these variants exist and have known for a couple decades now that there's a spectrum of structural variants in genomes. But the problem is that the reference catalogs that exist for these variant classes are relatively limited in sample size, especially in comparison to what we know about short variants. And so just to hammer home the importance of these reference catalogs a little bit more, and especially the sample size, I want to illustrate sort of a contrast. So take, for example, coding single nucleotide variants and coding indels. In 2018, which was sort of before we released the NOMAD structural variant call set, which I'll be talking about later today, the gold standard reference data set for coding point mutations or short variants was EXAC and or NOMAD, which was, depending on which data set you're looking at, on the order of tens of thousands or even over 100,000 individuals worth of data. And thanks to the public release and open release of site frequency data from projects like EXAC and NOMAD and others, we have witnessed in the last 5 or 10 years just an absolute deluge of advances across basic translational and clinical human genetics. And by contrast, unfortunately, structural variants are near and dear to my heart, but we as a field have really lagged behind short variants, painted as follows: that in 2018, the gold standard reference data set for population-based structural variant calls was still the 1,000 Genomes Project, which was derived from shallow sequencing in just 2,500 individuals. And although it was a really important advance at its time, in comparison to the large sample sizes we've amassed for coding short variants, I hope you can appreciate that the 1 to 2 orders of magnitude difference in sample sizes means that we really have not realized the same advances in our fundamental understanding of structural variants that have been facilitated for coding short variants thanks to these large population catalogs. This, of course, begs the question, why do we lack these large databases of structural variants if we can get them for coding short variants? Why can't we do the same thing for structural variants? Well, I think to answer this question, it really boils down to 2 key points. The first is that to comprehensively catalog structural variants at sequence resolution, you really need whole genome sequencing and deep 30x coverage whole genome sequencing. Structural variants can, of course, be ascertained with lower resolution methods like microarrays, and even, there's some methods now emerging for whole exome sequencing that look pretty promising for sequence resolved copy number variants. But to get the balanced rearrangements, to get things that are outside of coding sequence, which is the vast majority of variants, you need genome sequencing, and you need it in large cohorts. And of course, heretofore, up until very recently, this has been intractably expensive. And then the second, I think, is, given some of the themes of this seminar series, as I understand it, might be appreciated by this audience, is that the computational aspect of comprehensive structural variant discovery is a really hard problem, to the point where it makes you want to flip your desk. You get so frustrated by it. And I think the reason this problem is difficult is important to underscore. So there really is no silver bullet method, or there has not been. And even the methods, including ours, that have been developed in recent years are still imperfect. And the reason that they're imperfect is because it is a highly, highly, highly multidimensional problem. So unlike for a single nucleotide variant where you look at a single position in the genome and you just count the number of occurrences of reads that have the reference allele and the alternate allele, here, because structural variants are so multifarious and so diverse in how they present in actual human genomes biologically, you have to model multiple different types of sequence level evidence. You have to consider all the different types of structural variants that are out there. And like I mentioned, if we want to build a population catalog of these, you have to do this in thousands of genomes simultaneously, or tens or hundreds of thousands. And we're working towards millions of genomes. And so I hope you can appreciate that the genome is a big place, and we added many degrees of freedom to this problem. And so it becomes a data science nightmare, basically, to solve this at scale. So the vast majority of our work in the Talkowski lab and in partnership with many other groups around Boston, and specifically the Broad data sciences platform, has been on improving methods for germline structural variant discovery from whole genome sequencing. And I'm pleased to say that the official version 1.0 release of the product of our many, over half a decade of labor on this problem has been realized in the official release of GATK-SV, which is a cohort-based pipeline for structural variant discovery from germline genome sequencing data. And we have a individual case mode worth mentioning for those interested in analyzing individual samples that is still in beta, but it's publicly available in Terra as well as on the Broad Institute's GitHub. But that's not going to be the focus of my talk today. I'm going to be talking about the cohort pipeline. So the cohort pipeline is fairly complex, but I want to walk you through four main characteristics of it. The first is that given the multidimensionality of the problem that I just illustrated on the last slide, we maximize our sensitivity by running multiple orthogonal algorithms to try and capture as many structural variants as we can. We then reassess the empirical evidence in the sequencing data itself for every single predicted variant from all these different algorithms, which frequently disagree, many of which have high false positive rates. And so then we use a variety of machine learning tools to boost our specificity of the underlying calls by evaluating the raw sequence level evidence for each variant call that I mentioned. And then the whole back half of GATK-SV is a series of genotyping and harmonization and unification steps that integrates all of these predicted structural variant calls across your entire cohort and resolves complex variants. And so what you get at the end of this pipeline is a jointly genotyped and harmonized variant call file, or VCF, of all samples and all variants, structural variants discovered in your entire cohort. So turning to the Genome Aggregation Database, or GNOMAD, in the first release, which is the topic of today's presentation, we've applied GATK-SV to close to 15,000 standard 32x Illumina whole genome sequencing samples, of which a little north of 14,000 have passed quality control. And so just to put this into perspective compared to the existing and published public references for structural variants that I mentioned, you can see that this represents maybe a 6 or 7-fold increase in sample size over the existing references. So a pretty big boost. And while we are not capturing uniformly the global population, a majority of our samples, or 54%, are non-European genetic ancestry, which is a step in the right direction, and we're continuing to work on this for future releases of GNOMAD. So by applying GATK-SV to these 14,000 whole genome sequences, we discovered a total of over 400,000 structural variants across 7 different structural variant classes shown in the different colors here. And again, this is at least a 6 or a 7-fold increase in the total catalog, the totality of variants, structural variants known in population databases compared to things like the 1,000 Genomes Project or Genome of the Netherlands or the GTEx project. Translating this onto a per sample level, we see that we see a highly uniform burden of structural variants per germline genome in all unrelated individuals in GNOMAD, which is encouraging, suggesting that the method is working as expected. And it also is encouraging to see that we see patterns that match what we would expect based on population genetic first principles, like greater genetic diversity in individuals of African or African American ancestry, shown in the leftmost column, And in total, on average, across all populations, we're finding about 7,500 structural variants per genome. Then looking across all variants in the data set, we observe, not surprisingly, that most structural variants were small, meaning that methods and technologies that preferentially access larger structural variants are missing the vast majority of structural variants in the human population. And also that most structural variants are rare. So over 50% of the structural variants in NOMAD appear as singletons in just one individual in the entire cohort. And over 90% of all structural variants appear at a combined allele frequency across all populations less than 1%. Next, I want to talk about quality assessment. Unlike for short variants, there are no gold standard methods for benchmarking structural variant call sets from next generation sequencing data at scale. Of course, you can do things like PCR and Sanger sequencing for individual variants, but it's difficult to scale those, and computational and in silico methods are equally fraught for sort of the opposite reason. So instead, we applied a suite of 7 different approaches to be as comprehensive and agnostic as possible to assess the quality of the NOMAD structural variant call set. I'm not going to go through all 7 of these, but I do want to highlight 2 just to show sort of the variety of different approaches. The first is that in the NOMAD cohort collectively, we have 970 complete trios for which we have genome sequencing on the mother, the father, and the child. We can use this to measure a de novo rate of variants that appear sporadically appearing in the child genome that do not appear inherited from either parent. Given that we know that the mutation rate of structural variants is much less than 1 new structural variant per generation, we expect that the vast majority of these de novos are probably either false positives in the children or false negative calls in the parents. So this is sort of a proxy of a combined false negative, false positive rate. We see that's about 3%. So that is very encouraging, especially compared to existing structural variant computational methods. And then on the other side, we can do some molecular validation using orthogonal sequencing technologies. So using PacBio long-read sequencing, we were able to evaluate over 20,000 structural variants in a variety of selected different genomes because PacBio data is much, much more sparse still than Illumina sequencing. And using an in silico confirmation method that checks to see if the structural variant we predict in NOMAD is found in the raw PacBio long sequencing reads, we find about 94% of the time the predictions we make in NOMAD are found in their corresponding genome in PacBio long read sequencing, again suggesting a high positive predictive value of the NOMAD call set or specificity. And like I said, I'm not going to get into the details of all this, but you can find many, many more details in our paper and especially in the supplement. But the long story short is that all 7 of these approaches generally agreed that we emphasized specificity for NOMAD. So the call sets, the calls in the call set are by and large correct based on the methods that we can assess. But we do have a couple estimates of sensitivity, which we're encouraging, although sensitivity is much harder to assess for structural variants. Finally, in keeping with NOMAD as a consortium, our strong commitment to open data sharing and unrestricted access as a community resource, we've released all of the data I just described on the NOMAD browser. You can visit that at nomad.broadinstitute.org, and I encourage you to go take a look at that. I'm showing sort of a screenshot of how some of the structural variants appear, but this has really been spearheaded by the NOMAD browser team led by Matt Solomonson and Nick Watts. So shout out to them for really taking the data set that we produced on a research side and translating it into something that hopefully many of you will be able to find useful for your own research. So I'm going to pivot here and stop describing the data set and start talking about some of the analyses that we've done using the NOMAD data set to learn more about structural variants, getting back to the problem that I mentioned at the beginning of my talk, that our knowledge of structural variants is really lagging behind that of SNVs and indels. So one of the most fundamental properties of structural variants we still don't have a good grasp on is structural variant mutation rates. We know quite well for short variants how frequently they occur, even what trinucleotide contexts or nucleotide compositions predispose to de novo mutation in the human germline. And for structural variants, we basically know essentially nothing about this. We know roughly how many we see in children genomes in live births in the general population at a very coarse resolution, but we don't really know much more than that. And so we were able to use in NOMAD a classic population genetic-based estimator, the Watterson estimator, to come up with an estimate of the de novo mutation rate for structural variants across different variant classes in the population. This comes out to be about one new de novo structural variant for every 2 to 6 live births. This actually squares pretty well with an independent assessment that we did as part of a separate study where we predicted de novo structural variants in 519 quartet families, so 2 parents and 2 matched siblings, and we molecularly validated every single prediction we made in that data, every single de novo structural variant prediction we made in that data set. And so we can see that by and large, these predictions that we make in NOMAD match what we see empirically with molecularly validated data in quartet families. Another genome biology question we can ask is, we know structural variants can be complex, meaning multiple different mutational signatures interleaved into the same, compound or complex structural alternative allele, but we don't really know the spectrum of those mutations. And so in NOMAD in total, we really focused on complex structural variants as a specific sub-analysis and found over 5,200 completely resolved complex structural variants that averaged about 2.5 kilobases in size. And in total, we can break these down into at least 13 different mutational subclasses, of which I've collapsed a few here just because they're mirrored structures, like a deletion flanking an inversion versus an inversion flanking a deletion, which, in biology, in terms of actual DNA are pretty comparable, but in terms of the reference sequence are different. And so what we find here is that every single genome we've looked at has dozens of these complex rearrangements, many of which are rare, but given that the population in NOMAD is largely from unascertained selection from the general population with some important caveats, but certainly depleted of severe pediatric and Mendelian diseases, the evidence that we uncover here is that complex structural variants appear in every single individual's genome and by and large do not confer extremely strong risk for severe disease. And so keep that point in mind because I'll come back to that at the very, very end of the talk. Next, we wanted to ask the question, what are the functional consequences of structural variants that we can predict just from annotated genes in the genome alone? And so what we find is that structural variants contribute as many protein truncations, predicted protein truncations per genome, or predicted loss of function events, so PLOF is going to be the abbreviation I'm using here, as short variants. So each contribute roughly 100 to 200 protein truncating events per genome. And then when you focus in on rare variants, we find that structural variants contribute about a quarter of all rare protein truncating events per genome. And that is not even getting into the plethora of duplication, so dozens of whole gene duplication events or partial gene duplication events in every genome we looked at. So clearly structural variants are predicted to have a pretty substantial functional consequence on gene function. We can also look at this from a population genetic standpoint of measuring selection. So how deleterious are coding structural variants that we would predict would disrupt genes based on their annotated open reading frames in the reference genome. And so just focusing on 3 different classes of coding structural variants here. We have loss of function in red, and then 2 different flavors of duplication, gene duplication event in blue and purple, where the blue is a whole gene copy gain, and the purple is a partial gene intragenic exon duplication, which we would predict could potentially act more like a loss of function variant, but they're much more challenging to interpret based on context. We can look at essentially a measure of the proportion of each class of variants that appears as singletons, so in just one individual, compared to what you would expect based on the structural variant size, the evidence from our algorithm that contributed to that variant call, and a few other technical covariates that we know influence this measure. So this is APS, or adjusted proportion of singletons, and without getting into the specifics, if the value is greater than 0, that means that there is evidence of negative selection or purifying selection, that basically these variants are being weeded from the population. by natural selection. And so what you can see here is all of these colored dots are coding structural variants that are predicted to have one or more different consequences on gene function. And by and large, with the exception of one category, we see that genic structural variants are under, in some cases, quite strong purifying selection in totality across all structural variants in the human population. And note that we even see slight signals for promoter and intronic structural variants, which suggests that although they aren't as severe as loss of function events, for example, there is some non-coding effect that could be attributed to structural variants here. And I'll talk more about that in a second. And just as sort of a quality control or a sanity measure, I want to show you that this observation is also true even if you match within a given structural variant class. So for instance, looking at loss of function deletions in dark red versus their intergenic deletion counterparts in light red. And you can see that by and large, coding structural variants across all different variant types are under strong negative selection in the general population. We also want to look at how we can better interpret the structural variants that disrupt genes in a given genome. And so I want to focus on this concept of mutational constraint that many on the call today are probably familiar with, but just sort of a toy example to illustrate what this concept of mutational constraint means. Suppose we have two genes that are identical, gene A and gene B. They have the same properties, same number of exons, et cetera. When we look in a catalog like Nomad, we can see that gene A accumulates many loss of function single nucleotide variants in the general population, and gene B does not. There's, say, maybe only 0 or 1 or 2. By virtue of the fact that we see that healthy people in the general population have this gene inactivated, we can rule out gene A as being mutationally constrained, and reciprocally can say gene B, there's purifying selection acting on gene B to prevent it from being inactivated in healthy humans in the population. We can label that gene constrained. That's basically the general gist of what I'm talking about when I say mutational constraint for the rest of this talk. A major research focus for our group is to produce similar metrics of genic constraint and non-coding constraint for structural variants, so specific to deletions or duplications or inversions or insertions. But given the sample size that we're at of just a little over 12,500 unrelated individuals after quality control in Nomad, we're woefully underpowered to produce per gene metrics. This is a problem we'll solve with greater sample size, but until then, what we can do is we can use the existing metrics of constraint to evaluate whether we will expect similar metrics derived from structural variants to be informative as sample sizes improve. So what we did is we took all of the autosomal protein coding genes in the genome, so there's about 18,000 of those in the human genome reference, and we ranked them by their constraint against protein coding loss of function short variants, so SNV. So this is the constraint score from Nomad that I'm sure many of you have encountered previously or used in your own research. We then can essentially fit loosely predictive negative binomial regression model to ask for each group or each percentile of genes when ranked by constraint, do we see more or less structural variants in those genes than we expect by chance? For loss of function structural variants, we saw an extremely strong correlation between how constrained a gene is against protein coding, truncating single nucleotide variants and loss of function structural variants. This is, of course, not unsurprising given that we might expect shared modes of haploinsufficiency acting whether you're a structural variant or a PTV short variant. So this was an encouraging result, but what we saw that was a little bit less expected was that both whole gene duplications and those intragenic exonic duplications that I mentioned also show similar correlations with loss of function constraint, which suggests that these existing constraint scores from short variants that are much better powered than our structural variant data set are actually capturing a general intolerance to all rearrangements. And actually, I'm not gonna be able to get into the details here, but we show that this pattern does hold up for even non-coding structural variants like intronic variants or nearby enhancer structural variants, things like that. So we propose from this result that the existing constraint metrics are probably going to be useful for interpreting structural variants and not just deletions, but across a variety of different mutational contexts and classes. And then I also want to touch on, in a little bit more detail, the non-coding effects that I've been hinting at. So it's much harder to get at the estimating the non-coding functional effects of structural variants, given that we lack the exon intron annotations that we have for genes. We lack the trinucleotide codon cipher by which we can interpret those things. And so we're taking a fairly coarse approach here. So what we did is we took all of the strictly non-coding CNVs, so they do not overlap any protein coding gene whatsoever in NOMAD. And then just as a coarse approach, we compared them against 14 different classes of annotated cis regulatory elements or other interesting biochemical DNA marks like histone modifications, and then use the same adjusted proportion of singletons approach that I briefly explained earlier to measure selection against those variants. So if you take all of the intergenic CNVs, what you can see is that we see no real evidence of the average intergenic deletion or duplication in NOMAD being under stronger selection than you'd expect by chance. When you then compare that to the results that I've already presented for protein altering variants like loss of function deletions, and especially those in constrained genes, you can see that there's a strong signal of selection against deletions of constrained genes. Not surprising, we already looked at this. Next, when you split the non-coding CNVs in NOMAD based on whether they overlap one or more of the annotations of a broad variety of different classes that I'm going to show in a second versus those CNVs that overlap no annotated regulatory elements, you can see that there is a slight difference, and that but just by virtue of overlapping any annotated cis regulatory element, we see weak evidence of negative selection acting against those non-coding CNVs. And this is actually true across every single category of predicted element that we evaluated, ranging from everything from mammalian validated enhancer elements from the VISTA database all the way through large-scale computational predictions, and even just DNA's hypersensitivity sites from projects like ENCODE and Roadmap. And so this suggests to us that there is likely negative selection acting against functional non-coding structural variants in the general population, but importantly, that we didn't observe a single effect that was as strong as direct protein coding loss of function, which is again consistent exactly with what you might expect from population genetics, but it's really good to see that borne out empirically in data-driven analysis like this one. And then in the last 5 minutes, I want to highlight just a couple ways in which we think the NOMAD structural variant call set can augment existing disease association and clinical genetics. So the first thing is that we wanted to see if we could incorporate the structural variants from NOMAD into GWAS. And so what we did is we computed the linkage disequilibrium between nearby SNVs and indels in every structural variant, common structural variant in NOMAD, using in-sample LD for samples that we have both SNV and SV calls. And we found 2,300 in strong LD with one or more significant GWAS loci. Just one example of these is take this hypothyroidism associated locus in the UK Biobank. You can see a very characteristic pattern of linkage disequilibrium for this association. And when we overlap the NOMAD structural variants with this association, we can find a 336 base pair deletion of an ALU element that falls right at the peak of this GWAS association on the most associated haplotype. And this deletion, when we look at it functionally, falls in the first intron of a highly constrained gene. And this ALU element, which ALU elements are known to sometimes act as enhancers, is annotated as being active in adult thyroid tissues. This gene is expressed in adult thyroid tissue and GTEx. And in fact, the SNPs that are in this GWAS locus are also known to be EQTLs in thyroid tissue for this gene and a few other genes in the region. So I'm not saying that this structural variant is causal for the hypothyroidism association identified here in the conventional GWAS, but it is certainly worth, in our opinion, worth imputing into the GWAS and using these structural variant calls to help interpret and fine map the causal variants of these and other GWAS loci. And so, like I mentioned, we had over 15,000 structural variants that are tightly tagged by one or more SNPs that are commonly genotyped in most GWAS. And so the proposal that we're putting forward is, and we've released this data openly in the supplement of our paper, that you can take the nomad structural variants and impute them into your existing GWAS to help fine map complex traits and common diseases. And then flipping angles and talking more about medical genetics and rare variants, we looked at 3 different approaches here. So one is that there are a subset of sites in the genome for which large recurrent copy number variants are known to appear and are strongly associated with a variety of fairly severe, typically severe pediatric and congenital anomalies. These are collectively known as genomic disorder loci in the literature. And many studies using chromosomal microarrays, like in the UK Biobank, have tried to estimate the carrier frequencies of these in the general population. We did the exact same thing in NOMAD and showed that at sequence resolution, we see a very strong correlation with the frequencies of these genomic disorders in the population that has already been reported, suggesting that those existing catalogs are fairly accurate and that we will continue to improve the accuracy with sequencing. Next, we looked across a variety of different medically informative gene lists. So these are genes that are confirmed causes of dominant developmental disorders, or for instance, the genes from the ACMG that are worth considering for incidental reporting in clinical practice. And we just looked for very rare, so less than 0.1% allele frequency, loss of function events in these genes, in these dominant or recessive genes. And we found that, you know, a little under 1% of the general population, maybe say 1 in 200 individuals, is likely to carry, is likely a carrier of a rare loss of function event in one of these genes that has been flagged as being clinically relevant to consider if conducted in a genetic testing framework, which of course is a population screening study. We're just trying to benchmark the carrier rates of expectations of what we might find in the general population. And then finally, we wanted to investigate the hypothesis that very large structural variants are uniquely seen in severely affected individuals and in disease. And while large variants have been strongly associated with many different diseases and congenital anomalies, and so I'm not second guessing the legitimacy of that association, we do see that about 4% of the people in NOMAD carry a rare structural variant larger than a megabase, about half of which are balanced or complex rearrangements. So this is to say that if you do observe a large CNV in a genome, that does not necessarily mean that that structural variant is pathogenic by any means, even if large structural variants are strongly associated and more likely to be observed in affected individuals. And so just as an illustration of that, and as a final vignette before I close, and I'm happy to take questions and discussion, I want to give one example of just such a large and complex variant. So take, for example, this 49 breakpoint event found in a 56-year-old healthy adult sample in NOMAD with no prior indication of any severe disease. It was this individual was a control from a study of heart disease. So presumably any severe or Mendelian or dominant diseases would be a criteria for exclusion as a control from this study. This looks like an extremely complex event, but with genome sequencing and using GATK-SV, we were actually able to resolve this event into a 626 complex compound insertion rearrangement into chromosome 1. So what that means in practice is you can see all these different colored segments. Each of those was duplicated from a different chromosome, not chromosome 1. Somehow all of those segments were stitched together and shoved into a single position on chromosome 1. And so it's kind of like this chimeric large insertion, duplicated insertion, that when we functionally annotate this against known genes, we actually were surprised to find that we would predict no genic disruptions from this event, which may kind of explain the fact that, you know, how can a healthy adult carry such a complex rearrangement? Well, it may in fact be because of the fact that this fell into intergenic space on chromosome 1 and doesn't disrupt any genes. So there would be limited functional consequence we would predict at the gene level from this variant. So I think this is a really interesting vignette that emphasizes the value of sequencing large populations in a disease context, but especially outside of a disease context, to build these population data sets that allow us to observe these complex variants and learn more about what are the properties that confer disease risk and when can you observe them in healthy individuals. And so with that, I'll wrap up and leave time for questions. I would like to thank everyone that contributed to Nomad, and specifically the Nomad structural variant call set. Nomad has over 100 PIs that have contributed data and are part of the consortium. So there's far too many people to thank here, but I've listed some from the Broad on the far right, and in particular, Daniel MacArthur, who really spearheaded this and conceived of it along with Mark Daly and a few others from the very beginning. And then in terms of the structural variants specifically, I wanted to thank Harrison Brand, who's a new assistant professor at MGH and HMS, and was sort of my partner in crime in all things GATK-SV and Nomad related. And then our PI, Mike, who has been an excellent mentor and leader through this entire process. And so with that, I'm happy to take as many questions as there are. And if we run out of time, I've left my contact information down here. So please feel free to email me additional questions if we don't get to them today. Thank you.
Thanks for this extremely informative talk. I have a couple of questions. I haven't seen many questions posted yet, so please go ahead. So about the PacBio, what's the overall strategy of Broad or this consortium to run as much or as many PacBio confirmations as possible? What's the plan?
Yeah, so that's a really important question because it's shaping the direction of future sequencing initiatives like you mentioned. So where we stood for the initial release of Nomad, the data that I just presented, we were limited by the fact that long read sequencing was still in its not infancy, but sort of maybe elementary school years of scaling up. The number of, you can count on two hands the number of publicly available PacBio genomes that were accessible by anyone, at the time that we needed to do the confirmations for this study. So it really wasn't even a debate or a question at that time. Moving forward, there are many initiatives that are starting to sequence dozens or even some hundreds of samples with PacBio and Nanopore, and we've helped to develop methods for those, and it's coming along really nicely, but just the scale right now is the main reason why for Nomad, we are mostly interested in short read technologies, largely because we're currently processing 140,000 samples for the next release of the Nomad structural variant call set. We anticipate that'll be available early in Q1 of 2021, 2021 next year. And PacBio at that point, we think will be working with dozens or maybe 100 samples at that point. So eventually there's no doubt in my mind that PacBio will supplant conventional short read sequencing, but it really becomes a question of scale, and especially for population databases, the current answer is clear with short read Illumina genomes.
So when you mentioned PacBio validations, do you run a whole genome validation or a few cases, or it's more targeted?
Yeah, so we run, let me see, I think I have a supplementary slide on the PacBio specifically. I think this is actually the only one I have in here. But the study design is for the PacBio validations is basically, we have intentionally included a subset of samples for which genome-wide PacBio sequencing data has already been generated and Illumina short read. And so we're just able to take advantage of the fact that we already have two genomes with different technologies, and we can just line those up. And so for the next release of Nomad, we're going to have a few dozen examples. In this study at the time, we had less than 10 that we could really use, but we were still able to access, 20,000 variants for confirmation in just those few genomes.
I do not see many questions. Do you?
Yes.
See? Okay.
Okay. I have one question from the audience. Inheni, can you unmute yourself?
Yes. Hello.
Yes, we can hear you.
Okay. Thanks for interesting talk. I'm curious about how you calculate the early frequency of all the structural variant events in the Nomad SV. Like, it's very hard to imagine that two SVs found in two multiple individuals. I can imagine they will exactly match in terms of positions based exactly to the single base pairs. So I wonder how these early frequencies are calculated. Do you count like overlapping reasonably large proportions of two SVs overlap each other, then you count them as a same event and estimate allele frequencies like that?
Yeah, so that's a really good question, and coming from the standpoint of short variants, it can kind of be a little confusing and tricky to evaluate structural variant allele frequencies. I think one common misconception is that Illumina and short read sequencing doesn't provide base pair resolution, when in fact we see, actually it's perfect that I have this slide up. We see that over half of the variants in Nomad are resolved to within a single base pair. So on that basis, we can actually find direct sequence overlap of identical variants, presumably all arising from a single mutational event and not non-mutationally recurrent events, multiple mutations. So these are just population segregating variants, and for those you can just compute a very straightforward allele frequency, given that we've genotyped each of these variants and sites in all the samples. But what you do get at is a really important trade-off that we make in most structural variant pipelines, but certainly in GATK-SV, and that is that there is some sort of fuzziness around at what point do you consider multiple independently observed events, like let's take different large deletions that may not appear to have the exact same breakpoints. At what point do you consider them the same? The same biological variant versus different variants that just overlap a lot of the same sequence. And there we have optimized our clustering approaches to, given sequence resolved variants, like take, for example, PacBio data, where we know in multiple individuals, it's the exact same position of the breakpoint in reality. And if the Illumina estimates of those breakpoints are a little fuzzy, we can essentially titrate the amount of overlap that we require. And it ends up being largely based on the size of the rearrangement and what types of evidence contribute to that call. So if you have reads that directly split, short reads that split over a breakpoint, then you're in pretty good shape. If you don't, and you're relying on sort of anomalous pairs of reads, or even just a read depth signal, it becomes a little bit harder. So I think by and large, the allele frequencies, the computation of the allele frequencies is fairly straightforward, given a matrix of genotypes. But it certainly will, in let's say 5 or 10% of cases, be simplifying additional mutational complexity that we can't really access very well at scale with short read genomes. And that will be preferentially in regions of the genome that are known to be complex. So like short tandem repeats or segmental duplications and other low copy repeats, really, alpha satellites near centromeres and telomeres, things like that, where short reads just in our hands and with current methods really can't access the true complexity of the sequence under there. And actually, just on that note, we have a preprint up by Zhufeng Zhao, a postdoc in the lab, who did a direct comparison of sequence context and 9 different Illumina genomes and PacBio genomes, and tried to sort of disentangle where that lines up. And so just to make a long story very short, in the in the 92% of the genome that is not characterized by highly complex and repetitive sequence, we find a 95% overlap between PacBio deletions and Illumina deletions. But for insertions, irrespective of sequence context, we find that current long read methods, and specifically PacBio in the data set that we evaluated, just dominate sensitivity and specificity for novel sequence insertions especially. And so there, I think, are regions where our allele frequency and genotyping in Nomad are going to be not the most accurate. But for most deletions and large events in relatively clean genic sequence, I don't think there's a lot of cause for concern there.
I have a few more questions. If there are no more questions, I can't see anything here in the chat. So one of them is that the last case you presented, that's obviously very interesting. So how many cases like that did you see?
Yeah, so this is the most complex example. So of course it's selection bias that I thought this would be the most interesting example to look at. But in general, so if you look at the bottom right of this this slide, we find that about 1 in 200 samples carries a large rare complex event. And so by complex, our definition here is at least 2 different kinds of structural variant signature. And I think typically it will require at least 3 breakpoints, independent breakpoints. And this is outside of reciprocal translocations, like a simple P to Q arm swap, or very large inversions.
So are these, or did you try to look into whether these are perhaps chromotripsis events during meiosis, or what are these?
Yeah, that's an area of huge interest for us. So the more complex germline, I guess I should back up and say that for germline methods right now, we're not at the level of automated complex rearrangement reconstruction that a lot of the cancer methods are. Just because of the complexity of the rearrangements, we don't have a lot of good training data for that in germline genomes. We do have a few examples that appear to be chromothripsis or chromoplexy, but the complexity of those is relatively low. So like, I think we had a 12 breakpoint event with a few different chromosomes, things like that. By and large, the large rare complex events that we're seeing in NOMAD, and again, clinical sequencing of severely affected individuals is a totally different story. But in our studies in NOMAD and other population cohorts, we see very few but non-zero numbers of extremely complex events, like the one I showed on the last slide, like maybe on the order of 1 in 10,000 individuals, 1 in multiple thousands of individuals, I would say.
Right. Another question, if there are no more questions, so to what extent is your analysis biased when you consider low complexity versus high complexity regions? And I just can't imagine how you do that in repetitive regions or L repeats or whatnot. Are you considering those, or you're masking those out altogether?
So yeah, I mean, that's a huge caveat. And it's important to point out we are not masking them out by and large. I mean, there are some regions, like I think the centromeric regions, we just ignore.
Yeah, those must go.
Yeah, the alpha satellites are just not doable really with short reads. But for things like STRs and segmental duplications, and especially segmental duplications with 90 or 92% sequence identity, so more unique sequence in there, we do include them. We very often in those regions will not be able to capture specific breakpoints at nucleotide resolution, but for example, Steve McCarroll's lab and Bob Hansacker from Steve McCarroll's lab have been genotyping large multi-allelic copy number variants, which are wholly contained in segmental duplications for, the better part of half a decade now, and doing so with pretty good accuracy. And so we've adapted a lot of similar methods in GTK-SV, and we do capture, on the order of hundreds of large multi-allelic events in these highly repetitive sequence contexts. But where, to your point of like, what are the caveats? I would say that insertions, like I mentioned, is an area that we know we are not doing very well. We're not doing de novo assembly of the inserted sequences. We're not doing novel sequence insertions that don't appear in the reference. So insertions is an area where we're improving a lot for NOMAD v3, the next release that's coming up in the next year. And then beyond that, I would say that the genotype accuracy, the breakpoint precision in those regions just needs to be treated with more caution, I guess. Like, we can capture the evidence that there is probably a rearrangement in these highly complex regions, but we may not be able to piece together exactly what it is as well as we would in clean regions.
In terms of the first slide was sort of a size range on where you are operating right now, and you had the 50 base pair cutoff sort of, and actually we see a lot of trouble around that range. So sometimes variance between 30 base pair versus 70 base pair deletions, and they're not seen until we do a targeted sequencing. So are you good with that size range as well, or that's still very trouble? Okay,
tons of trouble. I mean, when you get sort of near beyond the size of a single read, but less than the size of a sequenced fragment, like a read pair, that is just really a nightmarish space that, short of doing a de novo targeted assembly, which we're working on methods with the Broad for doing that at scale, but even that type of analysis won't be in NOMAD v3. And so the average size of rearrangement in NOMAD SV is 300 some odd bases. That you start to see sort of a dip in the like 100 to 50 base pair range in total number of variants we observe, and that is almost certainly a sensitivity issue.
Basically, there's a blind spot still.
Exactly, yeah. still very much it's a problem, even with current methods for short reads.
So PacBio will solve that to some extent, I guess. At some stage.
Yeah. If you can capture the entire event in a single read, like in a PacBio or a nanopore situation, I'm really optimistic about that. And I think that's why we see such a difference in insertions in direct comparisons between, you know, Illumina and PacBio genomes for insertion sequence specifically.
Okay. I have one question. Sorry.
Go ahead. No. I could go on forever, so please go ahead.
Okay. So I have one question. So it seems like the constraint genes are quite important. So do you have this kind of gene list for what are constraint genes somewhere else we can find?
Yeah, so all of the constraint data are available for download on the Nomad website, and they're derived from each new Nomad release. So if you go to nomad.broadinstitute.org/downloads, there's going to be a link there for constraint data. And then there's a table generated by Konrad Karszewski, who was the lead author on the Nomad SNV and indel analysis, that contains a variety of different constraint metrics for loss of function and missense and recessive versus dominant and things like that for every gene that was annotated as a part of that analysis, which should be almost all protein coding genes. So you can download those data and play with that. And we found it really useful in our disease association studies, just as one anecdote.
Yeah, thank you.
Great. So if there are no more questions, then we are, getting to the end of the hour. So then we'd like to thank Ryan again for this presentation. And I'm sure that we all will get back to you at some stage for some help with the algorithms. So
Great.
Thank you very much again, and a great round of applause, virtual of course. And thanks again. Thanks.
See you guys on October 6th. We have a lot of seminar. See you guys.
Thanks.
Thank you. Thank you both.